Good afternoon, everyone. Swati, I'm not able to share my screen for some reason.
Yes, so one moment, please.
Thank you.
Can you please check now?
Yeah, I can do that now. Thank you.
Okay. Good afternoon, sir.
Thank you. Very good afternoon to you as well.
So, shall we begin this session or should we wait for a few more minutes?
Totally up to you.
Okay, people are still joining, so we can wait for 1 or 2 minutes.
Sure. In the meantime, I can check whether my screen is visible. So, can you please check if you can see full screen and you can see my laser pointer in red?
Yes, sir.
Perfect. Thank you very much.
All right, so welcome back, everyone. I hope you enjoyed the break. It is once again my honor to welcome Dr. Mohammed Asim, who will be presenting today on the topic metabolic vulnerabilities in cancer. Thank you very much, sir, for joining us. Over to you.
Thank you, Swati. Um, Likewise, pleasure to meet all of you today again. Um the structure for today is that we're going to talk about the metabolism, and especially with regards to the cancer cells. Sorry, my voice is a bit croaky today. Flu, not wanting to go, um, but hopefully I'm audible clearly. Uh, and in addition, what we're going to talk about later is some work, some research, latest research from my laboratory, uh, as well as from other laboratories on solving the problem of prostate cancer. So, without any delay, I'm going to talk to you about the uh, metabolism that is experienced by cancer cells and how you would start to appreciate differs from the normal cells. So have a look on your screen. What you can see here is basically central metabolic pathway, which is glycolysis. And then, of course, this is what we are taught in BSc. Um, Mostly those of you doing biochemistry know this pretty well. And then you have uh, Krebs cycle. And what happens here, the reduction of these NAD and FADH is then oxidized again through the oxidative phosphorylation cycle, which generates ATP. That's how the metabolism, as we know, works in normal cells. It has been noted that cancer cells are actually quite acidic. Where they are formed, the cancer is formed, they make sure that their surroundings become acidic. Now, why would that happen? Uh, why they are making and secreting acid in the nearby environment? One of the things which we now know is that it can actually stop the immune attack. As you know, cancer cells, because of the mutations, they express some proteins which are missing on normal cells. So these proteins are somehow changed in their shape thanks to the mutations. And that should make them visible to the immune system, right? But cancer cells can actually circumvent this. they make sure that even if they express these new proteins, what we call new antigens, uh, these are essentially coded by the genes which we already have. They're not new genes, but these genes, because of the mutations, have subtle changes within the protein structure that they give rise to due to mutations. And as a result, they should at least in theory become perfect target for immune attack. It does happen in some cancers, which are we were discussing day before yesterday, who have the MSI or microsatellite instability, something a pathologist or a genetic technologist in many countries can have a look. They can do karyotyping, look at uh, the chromosomes, but also electron microscope studies, and most importantly, sequencing will tell you whether there are microsatellite instability in some chromosomes. So this is very important. Cancer cells want to achieve a lot of things. They want to spread, they want to invade, they want to melt the matrix where they are present. And at the same time, now you realize that actually they are carrying out an altered metabolism so that they can enhance the production of acid, specifically lactate. And that allows them to evade the immune attack. So these cells are not really something which immune system will be able to see and attack them. And there are multiple advantages, which of course, these are textbook advantages. Uh, what I'll try to do is give you a brief overview of how it works and what are the key advantages to cancer cells, essentially tumors. I should also point out that when you're growing cells in a cell culture dish, you may not actually see many of these phenomena happening. The reason is very clear. Cells do not experience the same type of environment when you grow cells in cell culture dishes. You are growing just cancer cells. They don't have an immune microenvironment, but also they are not growing in 3D. They don't have any problem in getting oxygen supply. Why am I saying oxygen supply? Because as you would see that many of these pathways which allow cancer cells to secrete acid in their environment, they actually are carried out through glycolysis. And what happens in glycolysis is shown on your screen that cancer cells prefer. So normally what is happening here is that this pyruvate which is produced usually, and I'm talking about normal cells, it is converted into acetyl-CoA, goes into Krebs cycle. If a cell experiences that there is no oxygen or there is less oxygen, they actually decrease the flow of energy generation through this pathway, but they start relying on these pathways. This happen in normal cells, which cells, muscle cells, intensive aerobic exercises, we start getting cramps. And this happens because, as you would know, generation and accumulation of lactate. But what is happening here through generation of lactate, ethanol in other cell types, other organisms under oxygen depletion, is that you generate these acidic molecules in a cell. And as a result of this, what is happening are two things. One of them is that you don't need to generate ATP through Krebs cycle and through oxidative phosphorylation, which is, of course, a longer process. Uh, can you think of which pathway would generate more ATP? Do we generate more ATP through glycolysis or through Krebs cycle and then oxidative phosphorylation? The answer would be, of course, through Krebs cycle and then oxidative phosphorylation that generates much more ATP than glycolysis pathway alone. And it seems that cancer cells prefer this pathway, glycolysis. So looks like they're a bit silly in that they don't really use a pathway which is much more efficient. in generating energy from glucose molecules, but they rather rely on glycolysis. One of the advantages I just told you of you relying on glycolysis is that they can make their environment acidic. But there is something more happening here. I showed you 2 days ago a picture with birds during migration. Uh, they were prepared to do that migration. Exactly something similar is starting to happen here as well. These cells, when they're small tumors, they have sufficient oxygen supplies. As I told you the other day, tumors become bigger and they can actually try to invade, but also they can try to make new blood vessels to gain sufficient blood that can bring nutrition, right? So what these cells are trying to do is they're preparing themselves for an imminent lack of oxygen, because when solid tumors grow, many of them will not have sufficient oxygen, especially if you see, if you take a cancer biopsy and look at the internal cores, they will have very high hypoxia. They will not have sufficient levels of oxygen within them. And one of the reasons some cancers prefer to sit in metastasize to brain is within our body there is a different concentration of oxygen. Our lungs usually will have very high levels of oxygen. They are in direct contact of it, right? But as opposed to that, our brain has lower levels of oxygen. I'm not saying it does not, but if you look at the gradient, there is, of course, more oxygen in lungs than brain. Now, these cells are getting ready for a move. They don't, at this diagram, what you're seeing, they don't really need oxygen. They're trying to work out a metabolic pathway that can allow them to sustain without the need of using oxygen. Brain has less oxygen as well compared to lungs. And there is a whole gradient. You can have a look at that table. So what is happening is that these cells would also prefer, they would be perfectly fine sitting in brain and growing. And as we discussed yesterday, brain metastasis actually is super bad when it comes to the patient prognosis. So these cells look like they're very good in many aspects. They can carry out lots of acrobatic exercises. I was telling you, they move from primary tissue, they can go to other organs. Uh, but they're not very good at making use of glucose, it seems. Why? One of the reasons, again, is that they can make the environment acidic. And they also learn to live without oxygen. Uh, that is because they know eventually there will be a threat coming that will allow them to experience the deficiency of oxygen. So they're preparing for future like humans do, preparing for future. In this case, they're preparing for their future that they're going to experience deficiency of oxygen. So let's start not using oxygen and rather rely on this particular pathway, glycolysis, without the need of using oxygen. So if I show you an example, please feel free, by the way, to ask any questions if you have via the chat box, but I'll be more than happy to take questions afterwards as well. Uh, Swati, for some reason, it is saying continuous chat is off. So could you kindly switch on the provision that the attendees can ask me questions if they have any. So that's clear, hopefully. So have a look. What this is something which was noted by a scientist called Otto Warburg, that cancer cells actually use a pathway which does not really rely glycolysis, which does not really rely on the availability of oxygen. So have a look with regards to the energy generation, what is happening here. These are differentiated cells. You can think this of being part of an epithelial cell layer. And what is happening here is Swati. Can you hear me?
Yes, sir.
Okay, perfect. Yeah, I think the chat is off for some reason. So students or attendees may not be able to ask questions. They cannot type. Let me send a message saying
Chat is
test.
still on for the participants.
Okay, perfect. Thank you, Padmavati. Okay, so back to this topic again. So what is happening here is that normal cells usually they have good access to blood supply and therefore oxygen. And that allows these cells to carry out generation of energy, i.e. ATP, through the mitochondria, a Krebs cycle, and then oxidative phosphorylation, which generates a lot of energy per glucose molecule, as you can see on your screen. What is happening here that these cells, when they don't experience, I'm talking about muscular cells undergoing extensive aerobic exercise, they will not have sufficient oxygen available to them. And as a result, the system, the metabolism will be altered, leading to the production of lactate. Again, which is acidic. In normal individuals, it will cause muscle cramps, but in cancer, these cells are very clever. They're making sure that nothing comes and nothing that can destroy them, especially the immune cells. They will be deterred. They won't come anywhere near those cancer masses, seeing that they have their microenvironment. The environment where they're present is actually quite acidic. So as you can see here, this pathway is not very energy efficient. You generate far less molecule of ATP per glucose molecule. So if you see proliferative cells or tumors, there is something common here as compared to these differentiated tissues. So both proliferative tissues which are dividing very, very fast, perhaps at par with tumors, but then they are still normal tissues. Can you give me some examples of some tissues which grow very, very fast? These are the cells which are badly hit by chemotherapy. So the cells of our hair follicle, sorry, I don't have longer hair this time. So the cells in our hair follicle, they are one of the fastest growing cells, but also cells in our gut, they divide very, very fast. And therefore, you may notice patients, cancer patients who undergo chemotherapy will lose as a result of therapy. Chemotherapy will kill any cell which is dividing very, very fast. For example, tumor that is the intended effect, but they will also have side effects because they're also going to kill other cell types which are growing very, very fast. So fast-growing cells, especially cancer cells, what happens here is a very little of it, actually pyruvate, goes into mitochondria, and very little of it actually forms is gives rise to through oxidative phosphorylation ATP, but the majority of it is still making a lot of lactate. So it looks like cancer cells are not, after all, very, very energy efficient. They're rather very poor in this process. So if I show you some energy ratings of cancer cells, have a look here. They will perhaps be here. Their energy rating is really, really poor of cancer cells, thanks to their reliance on aerobic glycolysis. So they need, of course, they're growing much more faster than many. Sorry, they're growing much faster than many cell types, normal cell types, and at the same time, they're not making good use of glucose. So what happens as a result of this process? They can generate energy much more rapidly. Why? Because they're relying on something, glycolysis, which they can carry out in the cytosol. There is no need to involve mitochondria or the oxidative phosphorylation. So the generation of energy is much more faster. But also there are intermediates of glycolytic pathways which can actually take part in many synthesis reactions, anabolic reactions. Why? Because cancer cells are dividing much more faster than a normal cells. They will need a new fabric, they will need raw materials, and this raw material comes from many glycolytic intermediates. What I also mentioned is that by making and secreting lactate, it allows them to survive in a low oxygen environment, but also they can suppress the immune system attack through this. And lastly, but very importantly, what also happens when you have oxidative phosphorylation through the electron transport chain in mitochondria, that is the primary source of the production of reactive oxygen species. That is what is used by many therapies, including radiotherapy, to kill cancer cells by destroying the DNA. So if you can suppress the oxidative phosphorylation, that is what happens in aerobic glycolysis or Warburg effect. There is a less concentration of reactive oxygen species at this time, meaning the cells will survive this. They will not have very high levels of reactive oxygen species. I was telling you 2 days ago, there is something else which can also dampen down, which can also reduce the level of reactive oxygen species, which will be pro-cancer, which will promote cancer. Um, Antioxidants. So taking a lot of antioxidants in its super high, ultra high form uh, artificially may not be a very good idea for general population uh, themselves. So we should be very careful with that as well. I hope this is clear so far. So these cells are needing, they are growing much faster, but their system to generate energy is not very efficient. They are generating energy through glycolysis, but converting most of it into lactate, which is secreted. The rest of it is basically the source of uh, energy. You can see per glucose molecule compared to normal cells, they're making far, far lesser energy. But who needs more energy? Differentiated cell types or cancer cells? Well, cancer cells need more energy. So how do you compensate this? You compensate this by enhancing the level of glucose that you drink. So we know cancer cells are super hungry for glucose for this reason. They need to divide really really fast. They're not very energy efficient, so what they start to do is to steal glucose from their environment. So they become highly competent in taking up glucose from which was intended for other cell types. So have a look at this PET scan. What you can see here, under normal circumstances, we have tissues which are very intense. They have very high energy demand. And if you see them, these are the PET scan, FDG PET. So the glucose has been labeled, and that will show up on a PET scanner labeled glucose. And you can see here, if you do a PET scan of a normal individual, you can see here what cell types, what tissue types are actually the ones consuming the most glucose. So our brain, our heart, no doubt, as well as the transition, because of the transitional epithelium structure, which is very flexible, and the bladder seems to make use of a lot of energy. If I show you this slide, sadly, of a patient who has ARMS, the type of cancer of the alveoli, you can see here, in addition to the heart, brain, bladder, the cancer cells are actually lightened up by FDG PET. All these cells, what you can see here, they are consuming a lot of glucose. So this is actually what forms the principle of detection of cancer foci, not the primary tumor, but you can see even the little metastatic spread of tumor, where this is gone. Has it gone into liver? Has it gone into the pelvis area? Has it gone into the vertebra? And you can see, sadly, in this patient, it looks like cancer has actually spread a lot, including the bone system as well. So by exploiting the ability of cancer cells to take up a lot of glucose, you can actually help in the staging of cancer and localizing cancer as well, where is it located. And this information can then be used by radiotherapists to actually destroy through laser guided, with laser precision, where this cancer cells are and whether they can get rid of them. Uh, does it make sense? Any questions so far? Well, if there aren't any, then let's carry on. Okay, so what we discussed so far is the ability of these cells, cancer cells, to make use of a lot of glucose. Why? Because they want to do a lot of things. They want to make their environment really poisonous, i.e., acidic. They want a lot of glucose because their energy demand is far, far higher. Uh, and That's why they start to compete with other uh, cells and tissues, glucose, which was intended for them. And therefore, one of the things that happens to cancer patients is muscle loss, weight loss. Cancer patients will become really, really skinny. Many of them, in fact, one of the things that doctors, when they suspect in many countries, including India, substantial weight loss over a short period of time. Sometimes there may be suspicion of cancer taking hold as well, because of the fact that these cells are actually consuming a lot of glucose and therefore depriving normal cells of their diet, i.e., glucose. And in doing so, they actually cause sarcopenia, so loss of muscle fibers, muscles themselves, causing patients to lose a lot of weight. So they're using glucose as well, but as you would see, what can also happen in these cells is they become highly, highly metabolic, plastic. And what I mean by that is that normally what will happen, you can consider this, these are primary tumor cells, and they're sitting here on top of the basement membrane, and then they take a journey where they invade the connective tissue. They can intravasate to get hold of the blood vessels, and they go into the blood circulation system. We've discussed this before. I'm just revisiting it to give you a recap for the sake of this lecture. And what happens here, these cells, when they reach their destination organ, these cells can extravasate and start forming colonies in secondary organs, for example, lungs and liver. Bone, of course, is included as well. And what has been noticed that these cells, throughout their journey, they can actually change the requirement of nutrition. For example, these cells may rely primarily, while they are within this matrix, on glucose, but it has been noted that cells which are in circulation may have different need. They may change the requirement of the carbon source for as a nutrition. And you can see here they become more flexible in terms of the metabolic pathways that they can switch on, and therefore what nutrients can they use. So you can see, for example, nutrient 1, 2, 3, they can actually make use of them while they are in their journey. When they come back to their target organs, they may again switch back to using their original carbon source, which is glucose. As we discussed, all these cells, what you see, these black dots, is nothing but cells which are drinking uh, a lot of glucose, making the cancer foci visible on a PET scan. So if I show you the relative plasma levels of some metabolites, some carbon sources, have a look. Healthy individuals usually will have very low levels of pyruvate, but it has been noted as individuals develop cancer, their circulating pyruvate level increases. And in metastatic disease, in breast cancers and others, they have enormously high level of pyruvate, circulating levels of pyruvate. And same is true for lactate as well, especially in colorectal cancer. You can see an inverse correlation in glutamine and glutamate in esophageal squamous cell carcinoma. And what you're seeing here is there is a primary requirement of glutamine in primary cancer, but as cancer progresses, the requirement of using glutamine as a nutrition source may be dampened. And the opposite is true for glutamate. Normal or primary tumor cells will not have very much in the patients will not have in the plasma very high level of glutamate, but later stages, especially metastasis, will have very high levels of glutamate. And likewise, the similar is true for colorectal cancer and breast cancer and many other cancer types with regards to lipids. We know that one of these nutritional sources that these cells can use is actually none other than lipids. They can make use of lipids as a carbon source to generate energy from them. Uh, so these patients will also have very high level of lipids in the body. Now, what is the whole point of all of this? They're making use of these compounds. We are able to measure a handful of these metabolites in the plasma. There are people who are now thinking to detect cancer early, because if we know this full well, if you can detect cancer early, you can have better ways to treat it, and sometimes the treatment can be curative, meaning that cancer would not be likely coming back again, and they will die of some other cause than cancer. For that, we need to detect cancer early, and for that, what we need to do is collect samples from normal individuals uh, prospectively. So what has been happening, at least I know few trials, including the city where I live in, Cambridge, is that there are many GP services who allow you this to give you a sample of urine as well as blood. They ask you for 1 ml extra blood, and that will be uh, preserved. Same happens with urine. They will be able to freeze it and keep it there. What they want to do then, because they know that the population of the city is quite stable, there aren't very many people coming, and if people come, they usually live there for a decade or so. What they want to see is, okay, we are collecting the blood today, and we will follow up these uh, individuals to see how many of them develop particular type of cancer. So I'm not talking about 10 people on trial, I'm talking about thousands of people on these clinical surveillance studies to see which of them develop cancer. Then you can go back and have a look of the samples which was collected 20 years ago and measure the metabolite levels and see whether there are any patterns emerging with regards to the levels of these are 4 or 5 metabolites I'm showing, but actually through the chemical analysis you can measure hundreds of metabolites uh, in plasma samples and urine samples. This is exactly the intention, uh, because of which they're collecting a lot of samples these days to be able to go back retrospectively and test for whether they developed any cancer or not, and whether there were any anomalies, there were any metabolites, even 10 years, 20 years before the cancer became manifest on a scanner, for example, or manifested itself uh, clinically, that they can actually make any interventions going forward for future generations. So what we can do in scenarios like, of course, India is we have a big population that is positive. So if we can collect those samples from patients, not patients, normal individuals, and keep them. The challenge would be, of course, to keep them in -80 C, which requires a lot of energy itself. But if we can keep them certainly and start such clinical studies, we have a big positive, because the populations in cities is really very large, and even if you can get a fraction of people to take part in such trials, Actually, that would be very good in terms of detecting cancer early and perhaps make predictions for if somebody comes and we found out metabolite X was associated with colorectal cancer in younger patients, then perhaps those patients can have scans, endoscopies. Uh, they may be put on surveillance earlier if there is a strong association found with metabolite X and the diagnosis of colorectal cancer. And the cancer may be removed at a much earlier stage when it is a primary disease, really tiny tumor compared to metastatic tumor, of course. So that will certainly save precious lives, but also it will be, you know, good for you as research scientists to devise, uh, you know, protocols, clinical protocols like this that will allow you to do that. This is not something which can be easily done in Western countries, thanks to the decline in the population, aging population, but also the cost to the health economies. It's very expensive in these countries to even buy things like a simple proton pump inhibitor. I think 7 tablets you get is about 500 rupees equivalent. So there are good things in India that can be done, but of course, for that, basic scientists would need support from clinicians while they're taking blood, whether they're comfortable, of course, giving patients the information sheet, whether they would want to partake into clinical trials and give 1 ml of extra blood and a urine sample. So, of course, these are some things which we can do. Trust me, we have very good systems. We have good surgery and we have good intervention uh, for cancer. The problem with most cancers, which kill human beings, is that cancer is usually detected very late. For example, pancreatic cancer, it doesn't really say anything. There aren't any symptoms until it has a spread. And that's why cancer cells, for pancreatic cancer, the incident and the mortality is almost same. Within 5 years, it will kill 95% of the patient. So let me scientists have been able to devise molecules based on the ability of cancer cells or their hunger for glucose, small alterations in glucose molecule itself. So what you're seeing, this is a glucose molecule. What has been changed here by chemists is this hydroxyl is missing here. So now this is not glucose, but it's this 2-deoxy glucose, like deoxynucleic acid. This is 2-deoxyglucose. So what happens with this molecule? If you use this as a carbon source, of course, this is taken in by the cell through the glucose transporters and is actually recognized by the first enzyme, the hexokinase. And hexokinase can phosphorylate this and traps it as 2-deoxyglucose 6-phosphate. Well, hexokinase traps glucose in the same way. It phosphorylates glucose, so it cannot leave the cell. Likewise, what is happening here, 2-deoxyglucose can actually be formed by this reaction, 2-deoxyglucose 6-phosphate. Hexokinase does not really discriminate between glucose and 2-deoxyglucose, but once 2-deoxyglucose is formed, it can actually shut down this whole glycolytic pathway. So that is something you can think of using as a treatment for cancer, which are highly dependent, and in fact, majority of solid tumors, I said, are reliant on their ability to suck up a lot of glucose from the environment because of the Warburg effect. So experiments have been done with this 2-deoxyglucose where this has been given to animals, and I give you an example here. These are animals, mouse, in which the tumors are grown, and then they are given 2-deoxyglucose as a treatment. And look what happens here in these mice which have tumors. They can grow. You can see on how this is y-axis, the tumor volume, and how many days does it take for the tumor volume to for the tumor to achieve this volume. I'll start taking questions in a minute. I have about 6 minutes, then after questions. Khalida, please hold on. Uh, I've seen your question. So what is happening here? 2-deoxyglucose, when it is given to these animals, you can see here the tumor is completely flattened out. It does not grow as good as the tumors in the other arm, where there is no 2-deoxyglucose given. The If you take out those tumors after the study is complete, you can see here tumors in animals which did not receive the 2-deoxyglucose, the metabolic poison I showed you on the previous slide. These tumors actually grow much bigger within 2 weeks. But have a look here at tumors which were given 2-deoxyglucose. These tumors are really, really tiny. Mm, because of the fact that they are not able to grow as fast. They need glucose, so they will suck up 2-deoxyglucose thinking it is glucose, but in fact it is not. And as a result, you can see this can be achieved at least preclinically. The experiments that we do in animals, we call them preclinical experiments because they have not utilized any human being for the treatment. And for starting any clinical trials, at least up until now, for cancer therapy, you will need strong evidence that your treatment works in animals, at least two different species. One of them is mostly mouse, laboratory mouse. The problem with such therapies might come is whether these metabolic poisons are safe or not. There have been issues identified when these were given to animals. Sadly, uh, many times what people do when they're publishing papers, they only look at what I'm showing you on your screen. The growth of the tumor is affected or not. They don't see the welfare of mice. They don't even do, many of them, blood profiling to see whether there are any changes in mice. All they look at is the basic phenotypic changes, the behavioral changes in animals who have been put on these new drugs, but that's not sufficient evidence to take them into clinic. The drug regulators, clinical trials regulators, would want to see the data, extensive data of the side effects on animals before giving a green light to carry out such studies in human. And of course, the metabolism in human and a mouse are different, and because of that, treatments may not work in the same way. Does that make sense? So I will stop after this next slide I'm going to show you. This is quite interesting. Again, some changes happening into the uh, metabolic pathways, giving rise to new things. For example, if you have these aerobic glycolysis, you see that these cells become super hungry for glucose, right? You saw that in the previous example. In this case, what is happening because of some alterations, some mutations, to be precise, cancer cells start making some metabolites which are missing in normal human beings. One such metabolite is called uh, 2-hydroxyglucose. Now, what happens in Krebs cycle, if I show you here, especially this step, is that you have this isocitrate, which is converted into alpha ketoglutarate by the enzyme called isocitrate dehydrogenase or IDH. The mutations in IDH are one of the commonest in multiple cancers, especially glioblastoma, which is a specific type of brain cancer. Several years ago, I did some transcript analysis in prostate, and we found one of the top mutation and overexpression in the data set was none other than IDH. So IDH was mutated in cancer. Now, this looks quite simple. because of the mutation, the although the substrate is still isocitrate, the product which is formed through these reactions is not alpha ketoglutarate, but it is actually a slightly changed chemical entity. It is 2-hydroxy glutarate. Now, 2-hydroxyglutarate. So again, what is happening here? What the impact of this will be that the TCA cycle will be shut down or will be reduced. Why this is happening? Because cancer cells are trying to get ready for the imminent, for the near future loss of oxygen. They know that the electricity will be gone and they will be going in the dark. So get your torches ready. Likewise, what they're trying to do here, they know there is no oxygen going to be available. They cannot really rely on oxidative phosphorylation. They cannot rely on TCA cycle. So switch on glycolysis first thing first. They've done that. Okay, any residual acetyl CoA coming into the TCA cycle, block it. How do you block it? Block it by making mutations in the isocitrate dehydrogenase pathway so that you don't continue with this TCA cycle. In fact, make 2-hydroxyglutarate, which I'll tell you in a minute why this is important. What happens to 2-hydroxyglutarate? Uh, in terms of the fitness of cells under low oxygen conditions. It can block many enzymes. As you can see, many of them are epigenetic enzymes. I'm not going to go into the details of epigenetics, but 2-hydroxyglutarate is called oncometabolite. It is one of those metabolites which is not found in normal cells. And this is something which can be detected through the blood. So you really don't need a lot of tools in order to detect this. You don't need to do biopsies. You don't need to put patients under anesthesia or have multiple doctors operating on a patient. You can, in order to diagnose, you can simply detect it through the plasma. This is secreted by cells which have IDH mutations. The challenge for those of you who are a student is to identify more oncometabolites. Um, that's not what we're very good at. We just don't know what sort of metabolites are formed. Oncometabolites are formed by cancer cells. There are only few known, but nature is not silly. They're making them for some specific use. In this condition, so far what we have discussed is they're preparing for an imminent loss of oxygen, because as they grow, they're going to experience the loss of oxygen, and they're preparing it. They cannot rely on TCA cycle, so they're starting to enhance the level of aerobic glycolysis, what we call Warburg effect. They're shutting down this metabolic pathway, and then they're making this 2-hydroxyglutarate, which will help them again with the survival under conditions of oxygen deprivation. Who wants a break? I know I've gone through 45, 47 minutes. Dr. Aziz, shall we take a break? I know you're running late in terms of you're 5.5 hours ahead. If nobody wants a break, then I can continue to talk until I finish this. Totally up to you. Please let me know. Okay. In that case, I'll stop and see some questions which have been asked in the meantime. Those of you who want to take a break, you can take a break and let me have a look. Uh, so Khalida is asking, I've seen an old age person suffering from bladder cancer having all gray hair, undergoes chemotherapy as well as radiation therapy. After the treatment, he lost all his hair, but after some time when he hair grew, they were all black. What is the reason behind that? Really, this is news to me. I know that the people will grow back new hair. Uh, I don't know the reason why they grew totally black hair, but I will certainly check on this whether this is something that happens as a result of chemotherapy or radiation therapy. They do the hair do stop growing not as a result of radiation therapy. If it was given through EBRT, external beam radiation, that will not cause such side effects. These are the side effects which should come from being on chemotherapy. If they had bladder cancer, I'm sure they must have undergone treatment with BCG, uh, possibly. But if they were on chemotherapy, of course, they will lose hair for when they're on the treatment. They eventually grow back. I was not really. This is news to me that the hair was black. Any other questions, comments? You can switch on your mic. Feel free to ask me questions if you have any. Sandeep was asking, I came by a recent article where it was estimated how intermittent fasting is reducing cancer risk by starving cancer cells. Is it related to metabolic starvation or any other correlation? Very good question, Sandeep. So there are, of course, benefits known of metabolic fasting. Some of them, sorry, not metabolic fasting, intermittent fasting. Some of them, in fact, come from the ability of cells to undergo autophagy. This is very well known. A Japanese scientist received a Nobel Prize a few years ago on this discovery of autophagy being promoted under conditions of nutrient starvation. This is something we already know. Sandeepan, we have to realize humans, as we know, they were not really had uh, food like breakfast, lunch, dinner. We were wandering in jungles in prehistoric times. The food was not kept in freezers, was not sold in supermarkets, and it was not frozen. Of course, they had cancer, as I showed you, but perhaps the cancer was not as common as it has become now. And one of the reasons was that those people we know, they starved. They didn't have so good access to food as we do now. Yeah, with regards to intermittent fasting, we know there is certainly benefit of intermittent fasting. And we also know that mice, which are calorie restricted, from the same genetic makeup, if you have mice which are calorie restricted, sometimes live twice as much as mice which have been fed with high fat diet, for example. So we know calorie restriction does help in longevity, and definitely. Any other questions? Or we will start in about 8 minutes again. So feel free to ask me any questions if you have. Uh, well, plasticity simply, these are overlapping terms. But basically, what happens is metabolically plastics means that they're able to not only require, they are not absolutely dependent on one single nutrition for their survival, but they can use multiple sources. For examples, when we were living in jungles, what we ate as human beings, mostly roots, right? Those roots are really, really very super solid, very hard to chew. And you see many skeletons that we discover through excavations. Many of them have had problems with their tooth. 20-year-old male with many molars, premolars missing, trying to chew food. Some of them actually had a, we know that died of uh, infections, excruciating pain, going into their jawbone as well. Dr. Gupta can probably shed more light into this. But basically, we had a different food back then. Now our food is totally different. So you can see we are, as human beings, as organisms, we are quite metabolically plastic. What we can eat in terms of nutritionally plastic, should I say? So cells also, in order to become fitter, they can actually also use different varieties of nutrition as carbon source. And sometimes, because of these changes that they are going through, those changes make them fitter to survive, because you might find that some carbon sources are much more abundant in blood compared to solid tissue. So they learn to make use of this. And thanks to our pathways, metabolic pathways, which actually allow them to be used as well, because these nutritions, these carbon sources can enter through glycolysis pathway, also through TCA cycle as well. I hope that answers your question, Mustafa. Great. Dr. Aziz, just taking 4 more minutes for anyone to ask, and then we'll reconvene. Sandeepan is asking, there is a dietary concept of sugar cutting, and promoting natural sweetener like honey and jaggery, replacement for cancer patients and patients undergoing remission. But I see glucose is the main culprit of tumor cell promotion. Is it still valid statement? Um, Sandeepan, it's complex. It's an excellent question you've asked. And with regards to sugar cutting, certainly this might help. There are trials where patients have been asked not to have any sugar at all, especially the sugar with no nutritional value, what we call, you know, uh, only calories, pure calories, nothing else. With regards to jaggery and honey, of course, these are more nutritionally good. My suspicion would be, Sandeep, in the way our own immune system kills cancer cells is by many times generating these reactive oxygen species that need to be there for even cells during the immune attack. Uh, with super low levels, ultra low levels of glucose, how this is going to function. So these cells may actually be protected against immune attack. Maybe there is already some literature on this. I don't know. But with regards to, so many of these dietary interventions actually work more in settings of chemo prevention. Chemo prevention is something like chemotherapy, but chemotherapy you start after somebody has cancer. Chemo prevention, I would say, refers to something where there is still time for cancer to progress. You know that somebody in the family, the mother, father, developed cancer, perhaps died of it, sadly. So you can actually do this intervention 20 years before that, okay, this is very common in my family, prostate cancer, for example. Okay, I know epidemiological data suggests that patients who have very high dietary consumption of dairy products, they are more likely to develop prostate cancer. So those individuals may think of cutting dairy consumption from their diet as such. with regards to jaggery and honey, I do consume. Well, this morning I had tea with honey in it. It may not be suitable for many people to have a you know very high fructose diet. I would say I would not take into my body anything where there is no use of that substance. For example, these artificial sweeteners and so on. I'm not sure how they are metabolized by the cell. For glucose, I know there is a proper pathway which allows our cells to make use of glucose. Uh, Glycolysis pathway. We need energy. Cancer patients, of course, need energy. Sadly, most of the energy is being diverted to sustain cancer cells. But then it is a trade-off uh, what you want to achieve. So many of these interventions do work before the cancer actually, the diagnosis is made for cancer. As we discussed in the very first lecture, there are, of course, possibilities to decrease the chances of having cancer. About 70% of them are caused by our lifestyle. Hope that answers the question. Okay, so let's resume this session again. No problem at all. Okay, so you saw this. What is happening here? The cell is a cancer cell is trying to make some adaptations, and these adaptations simply mean that they don't rely on any metabolic pathway that requires oxygen. They learn, of course, to live with aerobic glycolysis, which gives them many benefits, as we saw. I know this is an energetically very bad process, but then there are lots of advantages of using this process. So, what can also happen theoretically, what you can do in laboratory experiments, we know that if you cut down glucose, like Sandeep, when you were asking about glucose, what could also happen anytime this is done? Glycolysis is inhibited, we know that the cells will try to adjust. And the way they do it is by enhancing the level of these receptors, the transporters, sorry, the glucose transporters. If you have more transporters, these cells will be able to attract more glucose. When you stop glycolysis, these transporters will go in number in order to try and compensate for the low levels of glucose. So one of the things that happens is this compound called glutar. This can be used to block these glucose transporter proteins. So the cells will have an imminent loss in the supply of glucose molecules to survive through glycolysis, and that will shut down their biosynthesis again. They will have low lactate, so they may become a very good candidate for attack by the immune system. But also, if you block this, what can happen is glutamine can be used as an energy source. And how that happens is that glutamine is converted into glutamate. Then glutamate can enter the TCA cycle via the alpha ketoglutarate entry point. So you can see in individuals who will have IDH mutations, they will have a deficiency of alpha ketoglutarate. But if you take a look at cells in culture which have deficiency of alpha ketoglutarate through IDH mutation, and you would expect to see alpha ketoglutarate is less, you may not find it. Why? Because it may be being supplied through the glutamine converting into glutamate and then entering the TCA cycle. So there are agents like glutaminase, this inhibitor CB839. So what can be done is you can block the glucose uptake by the cells using drugs like glutar, and simultaneously at the same time you can also block glutaminase through drugs, and that will have a very bad synergistic effect in terms of shutting down the metabolic pathways in cancer cells. You may ask, would it have any side effects? Well, on normal cells, yes, there will be some collateral damage. It may be that patients may not actually survive this kind of therapy and may experience some severe adverse effects, something that need to be checked. So have a look here what is happening in this example. This is lung cancer, and yesterday I was showing you one of the most prominent subclass of lung cancer. They have mutations in EGFR. We discussed this in some detail yesterday and day before. So this mutation in epidermal growth factor receptor, what happens here? These are very important in giving a signal transduction pathway green light to carry out this glycolytic pathway, which is important for lactate synthesis, as we know. It's also important for the synthesis of biomolecules, so anabolic reactions. uh, what can be done again, if you take this labeled glutamine, you can actually follow the fate of glutamine where this is going. You can also use uh, um the fluoro FDG, which is glucose, labeled glucose, and then see where this is going. You would notice if you're able to follow these radioactive ligands that many of them actually end up, the carbons end up in lots of biosynthesis pathways. So if you block EGFR using drugs like, this is a tyrosine kinase inhibitor, and lotinib, you can block this, and simultaneously you can use this glutaminase inhibitor to block this pathway, and you would see that this will actually have very pivotal synergistic effects on the growth of cancer cells. The problem again is, if you do any intervention like this, they work very well in cell culture dishes. Cells don't cry. You can of course look for markers like necrotic markers, DNA damage, and so on. But such therapies are something that may work initially very well in cell culture in vitro conditions, but as you progress through preclinical mouse models, these therapy may altogether fail. And that is why it is quite challenging to treat cancer, which is sitting in between our own cells. You can see the situation as someone having a flu or a bacterial infection. They have different pathways, and therefore you don't see very many side effects for many antibiotics. At least this is what we know so far. But for patients undergoing cancer therapy, which is again sort of a parasite like a bacteria, there is not very much we can do as much for as for a bacterial infection. And the reason is, this is the product of our own cells. These are eukaryotic cells of our own body, which have now stopped following the norms, the regular norms of life, and become actually rebellious. That's why it is very difficult to treat cancer. So now we will change gears, but please don't forget we are discussing these pathways with regards to cancer cells, their ability to try and adapt to low oxygen environment. And this will come again. This is actually very pertinent, sorry, to the new lectures. This one that I'm showing here on my screen. So what you're seeing here, one of the advantages which I did not tell you so far, is that the cancer cells, because of the altered metabolism, what am I meaning about with altered metabolism is none other than the requirement for oxygen is decreased. This allows cells to actually drive angiogenesis, and you can see here in some of these examples, the cancer cells, they're sitting here in the normal cells. These are tumor cells here. You can also see some cells undergoing angiogenesis. Looks like colorectal cancer. We'll have a look at this figure in the legend. So it looks like these cells are undergoing angiogenesis, making these new blood vessels supply, in order, of course, to make them gain more nutrition, but also at the same time to help them spread as single cells to distant part of the body. Why this is important? Because as I mentioned, benign tumors, they they remain very, very small. They don't really metastasize. They are not cancer. They don't have very many changes to their DNA. Malignant tumors, they're growing much faster. You can see these tumors. This is inches. Have a look at this. This is almost 2 inches, this tumor. Really a big tumor. Inside this here, they must be experiencing a lot of deficiency of oxygen because it's a solid tumor mass, um, sometimes calcified, very hard to gain access to. And you can see what happens here is they experience, you know, lack of oxygen. But this lack of oxygen, as I discussed previously, has some advantages. But one of the most important advantage is that it allows these cells to form new blood vessels. So anytime the, any cells will experience this lack of oxygen, cancer cells specifically will start to form new blood vessels. And one of the ways in which they would do it is by making enhancing the level of a protein. We call this protein vascular endothelial growth factor. I'm sure you all know about vascular endothelial growth factor. And we need vascular endothelial growth factor for our growth. When we are growing, our muscle system, our blood circulation system, blood vessels, they need to be elongated, they need to become larger. And we need, in injuries also, we need vascular endothelial growth factor in order to repair the injuries as well. So, what is known is that the production of vascular endothelial growth factor is, which is important for this to happen, new blood vessels to form. The vascular endothelial growth factor protein is formed more by cells when they experience hypoxia. So when the cells are experiencing hypoxia, I discussed you previously, they're relying, they're learning to rely on using glycolysis. They're trying to stop TCA cycle. They're trying to stop oxidative phosphorylation, which will kill them through the generation of reactive oxygen species, uh, through autophagy maybe. So they are trying to block those pathways, but also through the lack of oxygen, they are triggering the expression of vascular endothelial growth factor protein. The conditions of low oxygen, we call them hypoxic conditions. There are agents which have been used, there are drugs, experimental drugs, which have been used as inhibitors of vascular endothelial factor. If you block vascular endothelial growth factor, the cancer might just remain at one place, may not spread at all. If it does not spread, it's pretty easy for most tissues for doctors to remove them, right? But the problem with cancer is that it spreads, it metastasizes, it invades in the nearby organs, it spreads through metastasis to distant organs. That's the biggest problem, right? So vascular endothelial growth factor is something that is very important in driving synthesis of new blood vessels, a process we know as angiogenesis. So let's see what happens when our body, and I'm not talking about cancer cells, I'm talking about our body experiencing hypoxia. We have less oxygen in our body. So one of the causes of having less oxygen in the body will be what brings oxygen to the lungs in the first place. Um, what happens to the oxygen that is taken to tissues, how it is taken to tissues through the RBC. We know that. What happens is that there is an important hormone that regulates the formation of RBCs. We call this hormone erythropoietin. Now, erythropoietin is a type of growth hormone, which is again a protein. It is made exclusively by the cells. You see these cells in the cortex, the outer cells. These are the cells which are making the hormone erythropoietin. Now, what happens is there are patients in which As a result of chronic kidney disease, you have patients who have compromised kidney function. Uh, One of the bad things happen to them is low oxygen levels, low RBC count, and that is a result of destruction of kidney. If kidneys are damaged, they will not be able to produce sufficient erythropoietin. No erythropoietin means no RBCs simply, and less oxygen. Brain cannot survive without oxygen. It needs certain level of oxygen to survive. We all know that. So why am I telling you this? I'm just telling you how our body copes under physiological conditions when it experiences hypoxia or low levels of oxygen. And this is also relevant to cancer cells because they are, after all, product of our own body. So what happens? Have a look under normal conditions when oxygen is not deficient. There is plenty of oxygen. That only the specific cells in this region, cortex, they are the ones in the kidney who produce erythropoietin. You have less oxygen, RBC go down, of course, the expression goes up. What happens if someone is experiencing extensive hypoxia or individuals going on high altitude, for example, where oxygen is less, or individuals, of course, in cancer, as a result of hypoxia, the system, the body will try to counter the effects of hypoxia. In normal conditions, in normal individuals, red dots, not only the cortex cells, but also these medullary regions of kidney, they start to produce enormous amount of erythropoietin, as much as over 100 times amount of erythropoietin compared to these conditions. And that erythropoietin is going to send a stimulus to the bone marrow to make more erythrocytes or RBC. More erythrocytes will try to capture more oxygen and try to settle the condition so that the person returns to normoxia. I hope that makes sense. So, not only human beings experience hypoxia, as I mentioned, cancer cells do as well. And I'll show you a pathway which is regulated by a transcription factor. And what this pathway does, in fact, do is to drive the expression of certain genes to help those cells survive the effect of hypoxia. Like these cells, what is happening here? There are cells which are making erythropoietin. But what is that pathway which is allowing them to make this erythropoietin protein, which is a hormone that triggers the synthesis of RBC? So you can imagine this pathway. The kidneys, what happens when they start to make and secrete erythropoietin, you can have more RBCs. But this is for, in order for kidneys to do that, they need to experience low oxygen. When the oxygen concentration decreases in conditions like anemia, most people will have their kidneys, if they're intact, if a person does not have a chronic kidney disease, usually kidneys will be able to enhance the level of RBC production through the synthesis of erythropoietin. The question is, what is making this erythropoietin? What is the proteins upstream of erythropoietin? What is the signaling pathway that actually does that? Have a look here at this pathway. So again, what you're seeing is a cartoon. And in this cartoon, you're seeing here a cell, a eukaryotic cell, and the nucleus. In the nucleus, there is DNA. Well, we saw that yesterday with the example of P53. We saw that yesterday, day before yesterday, with P53, and with yesterday with the example of androgen receptor. Look what is happening here. There is, like androgen receptor, there is a transcription factor, and this transcription factor is called HIF1 alpha. What happens under normal conditions? HIF1 alpha simply stands for Like androgen receptor, or AR stands for androgen receptor. HIF1 alpha stands for hypoxia-inducible factor. This is a protein made by cells when they experience hypoxia. And this is a protein which is also activated in kidney cells when they experience hypoxia. So what happens under normoxic conditions, meaning there is no shortage of oxygen at all, there is plenty of oxygen in the system. In these conditions, these oxygen molecules will bind to the HIF1 alpha through a covalent interaction. They don't bind just like that. They need an enzyme called PHD, which carries out this reaction, which carries out prolyl hydroxylation. So under normal conditions, in a cell, you will not find very much HIF1 alpha protein if you do a Western blot. Simple answer is HIF1 alpha is degraded. It is not there. Why? Because HIF1 alpha can be hydroxylated by prolyl hydroxylation, and then this hydroxylated HIF1 alpha becomes a substrate for VHL, which is a protein which, if it acts on certain proteins, any of its substrate, once it acts on those substrate proteins will be degraded. Their destination will be taking them to the abattoir and dying. Those cells will chop off through the proteasome those proteins which become the target of VHL. This is a simple E3 ubiquitin ligase reaction. So now, to tell you in another way, what is happening here, in normal conditions, HIF1 alpha function, which is a transcription factor, is not needed. When it is not needed, it is degraded. Of course, in normal conditions, you will have plenty of oxygen inside a cell, which will promote this reaction, right? The enzyme that carries out this prolyl hydroxylation is called PHD. Uh, that's the name written here. That's the enzyme causing prolyl hydroxylation, and that will degrade this. Do you know the cofactor for prolyl hydroxylase enzyme? This enzyme? The cofactor for PHD is the same protein, is the same metabolite, which is an oncometabolite. Can someone tell me what is the oncometabolite we discussed, which is produced as a result of mutant isocitrate dehydrogenase pathway? The name was not alpha ketoglutarate, it was 2-ketoglutarate. So, what is happening here? The same alpha ketoglutarate, the product of a wild type isocitrate dehydrogenase reaction, that same enzyme, but wild type enzyme, gives rise to alpha ketoglutarate. If you want, I can show you that reaction again. Uh, have a look here. Isocitrate in TCA cycle is converted into alpha ketoglutarate through the normal IDH enzyme. But if you have mutation, this IDH enzyme mutated will convert isocitrate into 2-hydroxyglutarate. What is happening here? These cells with this mutant enzyme will experience shortage of alpha ketoglutarate being synthesized through the TCA cycle. We know that now. So the same alpha ketoglutarate is needed by the PHD enzyme to become active. Once PHD enzyme becomes active, what will happen? HIF will be degraded. There won't be any HIF. There will not be any synthesis of genes which are needed for cells to survive in low oxygen conditions. Does that make sense? I hope it's not complicated. So 2-hydroxyglutarate is something which is produced through the mutant reaction, but in normal conditions, the wild type isocitrate dehydrogenase is making alpha ketoglutarate. So what is happening here? This enzyme PHD requires alpha ketoglutarate to become active. And what is happening? The alpha ketoglutarate will not be there. Why? Because there is a mutation in isocitrate dehydrogenase. What will happen as a result? The HIF will not be degraded. the HIF will become active. So we know what will happen once HIF becomes active is that the HIF will not go through this pathway. Under hypoxic conditions, there is no oxygen anyway. This PHD enzyme is not active. The HIF will translocate into the nucleus. And remember, P53 was binding as a tetramer to the DNA. Androgen receptor was binding to the DNA as a dimer, as a homodimer, two same androgen receptor binding to the DNA, driving transcription. In this case, what is happening if an alpha pairs up with ARNT, or if one beta, as we also call it, binds with this second protein, heterodimerizes, bind to DNA. Now you will ask me, so what genes does it regulate? Well, one of the genes that it regulates is erythropoietin in kidneys. Once this HIF1 becomes active, it enhances the level of erythropoietin expression that leads to the formation of these RBCs. So this erythropoietin being formed in enormous amount is because of the HIF1 alpha becoming active, as is shown here. The other protein, which is very relevant to cancer cells, which HIF1 regulates, and there are many, many proteins, in fact. Um, I'm just giving you some very simple examples. The other protein, one of the most important for the cancer cells to spread themselves throughout the body, this protein is vascular endothelial growth factor. VEGF, vascular endothelial growth factor, is also under control of HIF1 regulation. So HIF1 alpha, as a transcription factor, once activated, can also increase the level of VEGF in cancer cells. You can see here in normal cells, HIF1 alpha is degraded by the proteasome pathway through this reaction that we have discussed, the hydroxylation of the proline residues. There are specific protein residues within HIF1 alpha which become hydroxylated through this reaction, prolyl hydroxylation reaction. So what is written here is simply what I discussed. So what happens here, in a sense, is you have this tumor-associated angiogenesis taking place in tumor cells experiencing hypoxia, in kidney cells experiencing hypoxia. The HIF1 alpha will selectively enhance the level of erythropoietin. That's what is needed in a normal individual. But in cancer cells, they're also experiencing hypoxia. And once they experience hypoxia, they secrete vascular endothelial growth factor molecule. And this vascular endothelial growth factor actually stimulates, it drives the blood supply into them, so that these cells, the cancer cells, which are sitting in a primary tissue, can now actually spread to distant organs. So what scientists are trying to understand, of course, they see this as an opportunity. The cancer cells, they have a very uh, low levels of oxygen. They experience hypoxia, while normal cells have plenty of oxygen. So they are trying to find drugs, interventions, which will specifically target and kill the cells experiencing hypoxia, meaning the cancer cells. And have a look at this molecule. Sorry, is someone asking someone? Kavish? Okay, no worries. So you can see this molecule TPZ, which has been synthesized. What happens with this molecule? This molecule specifically becomes reduced in cancer cells, or the cells which are experiencing hypoxia. And this reduction of this molecule is indeed required to make it active at killing cancer cells. So this is now actually, this is a prodrug. This is an active drug. And one of the things it does is generate these hydroxyradicals, which will kill tumor by destroying the DNA. So these hydroxyradicals are formed. This will be very important. So the drug, although given systemically, perhaps like a tablet, it is only going to become active in cancer cells because they're experiencing extreme hypoxia. They're growing very, very fast. I'm talking specifically about cells which are solid tumor cancer, um, experiencing hypoxia and will form these radicals. In order to destroy tumors, normal cells may be spared, may not experience as detrimental side effects as the cancer cells will. So with this, I can summarize this topic that we are discussing. The bioenergetics alteration in cancer cells is actually sits at the heart of these cells, which is important for cells to survive in a low oxygen environment. You can see how many things a cell is trying to do. This is super important for cancer cells to be able to survive in low oxygen environment because nothing, as we know, the life in primitive times when the earth was near, newly formed, the environment was reducing, right? That is what the scientists believe. Um, Later on, it changed to oxygen. But those conditions, if created now, we will all die, except, of course, some bacteria which can make use of those conditions is still found in Earth, on the Earth. But otherwise, oxygen is so much required. But these cancer cells are excellent engineers in making sure, in training themselves to make sure that they have right pathways. So changes in the central metabolic pathway, not relying on DCA cycle, not relying on oxidative phosphorylation, which can kill them, relying mostly on glycolysis, trying to block the DCA cycle, trying to activate the HIF. All of it is they're doing in order to survive the conditions of extreme low oxygen tension. And that is why this altered bioenergetics in cancer cells is so important. We saw this oncometabolite, one of them, this 2-ketoglutarate is very important, but one of the things it is